In this figure, we're going to talk about cell-cell junctions. And we're going to use an epithelial layer of cells here as our example where we find many cell junctions. Now, a couple key things to point out is that epithelial tissues do not generally express or necessarily express all types of cell-cell junctions. We're just showing them all in one layer of cells here. And also, we're utilizing epithelium as a good example of cell-cell junctions, but it's not the only one. Cardiac muscle, skeletal muscle, smooth muscle, and a variety of other tissues will demonstrate cell-cell junctions. And we use epithelium only because it's such a cell-dominated tissue that it's a good example. Now, those cell-cell junctions are going to fall into three main functional categories. There's going to be a category for communication, enhancing communication between cells. There's going to be one for mechanical attachment of those cells, and one about regulating the permeability between these cells. And let's look at these in reverse order. We start with the permeability cell-cell junction, and that is the tight junction. And tight junctions are formed by claudin and occludin proteins, and they form an intercellular mesh here. This mesh in the paracellular space, the space between the adjacent cells. And its function is to create a barrier to passive diffusion across that paracellular space. This limits permeability across this layer of epithelial cells. Now imagine if this were the GI tract and we had nutrients out here that we were trying to transport into the body, right? We would be moving those in a transcellular pathway across the cells, out into the interstitium underlying this epithelium and into the bloodstream. But as those solutes accumulate here in the interstitial space, they would have the potential to diffuse backwards. These tight junctions limit that paracellular permeability and make sure that we end up with a largely unidirectional movement of the substances here. This is also true in the kidney nephrons where we're trying to put wastes into the urine and we don't want them diffusing passively backwards. In our second category of function, we have the mechanical attachments. We're going to look at the desmosomes and the hemidesmosomes. Now, the desmosomes utilize cadherin proteins, and their function is to link cells to each other in a structural fashion. In other words, creating mechanical connections that keep these cells from coming apart. We can't just link the membranes of adjacent cells. The membrane is a very flimsy structure. And so these desmosome proteins are linked to intracellular cytoskeleton proteins, shown here in red. And as you can see, we could have a desmosome linked to the cytoskeleton, linked to desmosome, linked to cytoskeleton, linked to desmosome. And so what we're seeing is this mesh of proteins that span the layer of cells, creating a structural framework such that all of those cells are sort of tied together. This becomes obvious with cutaneous membrane when we get a sunburn and we kill a whole bunch of these cells all at once. Well, when those dead cells die, they come off in big sheets, all tied together by these desmosomes. Now, the hemidesmosome is also an anchoring junctional complex, but in this case, it's going to utilize integrin proteins, and it's going to tie the cell not to another cell, but to underlying connective tissues, in this case, of the basement membrane. All epithelia are attached to a basement membrane, which is secreted by both the epithelium and then the underlying connective tissue found underneath here. And so what this does is it prevents this layer of epithelium, in this case, from peeling off of the underlying tissues. If that were to occur in our cutaneous membrane example, what would happen is that cutaneous epithelium, the epidermis, would peel off of the underlying dermis, resulting in extensive blistering. So once again, we want to attach this epithelium strongly to the substrate beneath it. Finally, we're going to talk about our communication cell-cell junction, and that's the gap junction. In this case, the proteins are called connexins. They form complexes called connexons, which are essentially pores that are formed in each of the adjacent cells. And when those pores dock, what happens is we connect the cytoplasm of those adjacent cells, allowing diffusive flux of small solutes, including ions, that could carry electrical current. This results in the formation of what's called an electrical syncytium. These cells are now electrically connected and can function as a unit because electrical signals that coordinate the activity of those cells now link them together. These electrical signals travel rapidly cell to cell, allowing us to create a functional unit of cells known as a functional syncytium. This is enhanced by the presence of desmosomes in the heart, where those cells might not only need to be electrically connected, but also mechanically connected because they're pulling against one another as well.